Welcome to our biology class, and in this class I will be explaining on meiosis. So it's just a continuation from where I ended in part 3. So when you talk of meiosis, this is a type of cell division that normally occurs to produce the germ cells, just like I explained in the previous class. So when you talk of the germ cells, we are simply talking about the sex cells, and these sex cells, we are simply talking about the egg cell, which is also known as the oocyte or the sperm cell. So with that information, you need to also take note that daughter cells which result from the meiosis, they normally carry half the number of chromosome and half the amount of genetic as compared to the parent cell. And in most cases, this type of cell division, it leads to the production or to the creation of 4 daughter cells. So there are normally 4 daughter cells which are produced during the process of meiosis, and each genetic information or the number of chromosome in every cell which is produced, it's half the amount of cells, half of the amount of chromosome which are in the parent cell, and that half amount of chromosome is known as the haploid number. So in meiosis, the produced cells have an haploid number of chromosome. Okay, so meiosis is also known as the reduction division. So because of that reduction in the amount of chromosome from diploid to haploid, it is the one which leads to this process being called a reduction process. And this meiosis occurs during the formation of gametes and the process which is known as the gametogenesis. So during the process of gametogenesis, in female it is known as oogenesis, and in male it's known as spermatogenesis. So during the oogenesis, you need to understand that the major thing to be produced is the sperm cell, and then during the oogenesis, the oocyte, which is the egg cell, and then during the spermatogenesis is the sperm which need to be produced. But at the end of each process, whether it's oogenesis or spermatogenesis, they need to be 4 daughter cells. They have to be 4 daughter cells that are going to be produced. But there is a difference between gametogenesis in male and gametogenesis in female. So when you study embryology and human anatomy, you'll be able to understand more about what happens and the difference. So characteristics feature of meiosis that the nucleus normally divide twice. And this simply means that meiosis is a process which is divided into 2 categories. You have meiosis 1 and the meiosis 2. So let's quickly move on and look at I think this diagram, I explained it in the part 2 of this same topic. So let's quickly look at meiosis division. The first meiotic division, this is also known as the reduction division, and at this during this stage, the number of chromosomes are being changed from diploid to haploid. So it is during meiosis 1, that's where you need to expect the chromosome to be what? To be reduced from diploid to haploid. For more explanation, I think I delivered in part 2. Then we have interphase, which I already explained in part 2, and then this diagram I also explained in part 2. Now, let's quickly look at meiosis 1. I'll start with prophase 1. So this is where I ended, and this is where my explanation will start from as of today. So during prophase 1, DNA is exchanged between the homologous chromosome. So in some lecture tests and also the final exam, they might put sister chromosome, which is wrong. So you need to understand that DNA is exchanged between homologous chromosome in a process called homologous recombination, and this normally takes place or result into a what? Chromosomal crossover. So when you talk of chromosomal crossover, there is normally the exchange of DNA between the homologous chromosome, and this happens at prophase 1. The other thing that you need to understand is that the new combination of DNA created during crossover are a significant source of genetic variation. Like I said, even in families like you and your siblings, you don't look the same. You don't look exact the same. Despite of the fact that you are both coming from the same parents, your mom and dad, they are just the same. But even that, you still have that variation in terms of how you look, because your phenotype, which is how you look, it depends on the genotype, which is the inside genetic information that you have. There are certain things that we cannot change on us, which is our genetic information. It's determined by the DNA which we inherited, the genetic information we inherited. For example, I cannot change how I look. I might be looking dark in complexion. I can't change that. Okay, that is a me. That is my genetic information, which is showing outside as a phenotype. Even if I was to use the breaching agents, that breaching agent will just try to change how the melanocytes behave. The melanocytes are the cells which produce melanin. So these melanocytes, they will start releasing few amount of melanin. But once I stop breaching, my skin will start going back to normal. Why is it going back to normal? It's because of what? Of the genetic information which I have carried. Even how I talk, it's something that is genetically inherited. Some people, even how they behave, some are very quiet, some are very active, some are good public speakers, others are not good public speakers. It's just because of the genetic information which they have. Though some of the characteristics can be changed depending on how someone was brought up. So during the same prophase 1, you need to expect the paired and replicated chromosome, which are called the bivalent. So a bivalent is formed during the stage of prophase 1, which is known as the zygote. And then we have the tetrad, which is having 4 chromatids, and for this one, it normally takes place at lactotin. Now, the process of pairing the homologous chromosome is called the synapsis. So all this will simply take place at prophase 1. Take note of the definition, synapsis. Now, at this stage, the non-sister chromatid may cross over at points called the chiasmata. So there is some genetic exchange during crossover, and this is due to the fact that the sister chromatids, the sister chromatids are held together at the at the chiasmata, so they can change the genetic information. With this in mind, we can move on and go further in detail what prophase 1 is. So prophase 1 is the longest and involves a lot of series, and these series are being divided into 5 stages. So there are 5 stages that makes up prophase 1, and this prophase 1 is the longest amongst them all. Okay, so with this information, let's look at the stages of prophase 1. There are 5 stages of prophase 1. Number 1, we have the lactotin. So during the lactotin, you see the chromosome become visible. So in most cases, you are able to see the chromosome at lactotin level. Then you have the zygotin. That's where you're going to expect the formation of the bivalent, and you're going to have the pachytin. That's where you can see the tetrads and also the exchange of the same thing. Then you have diprotein. The chromosome will start dividing, and lastly you're going to find the dikinesis. So let's now go in detail and look at each of these guys. So let's quickly look at lactotin. At lactotin, the chromosome will shorten and become visible as a single structure. So you are even able to see the chromosome at lactotin. That's why chromosomes are visible at prophase. Zygotin is the next stage. So at this stage, there is paternal and maternal chromosome coming together and they pair up. So the pairing of homologous chromosome is called synapsis. So synapsis takes place at prophase 1, and the stage is what is zygotin. Now, the paired homologous chromosome are described as the bivalent, and this bivalent, it normally shortens and thickens. That process where the bivalent shortens and thickens is known as spiralization. Okay, so from there we have pachytin. So each chromosome will split into 2 chromatids. So the homologous chromosome which we experience at zygotin, it will split into 2 chromatids, and due to this splitting, it will lead to the formation of what we are calling a tetrad. So a tetrad has 4 chromatids, of which 2 are from the male and 2 are from the female. Paternal means male and the maternal means the female. So these 4 chromatids, they are going to form a structure which is known as the tetrad. So such will be experienced at the pachytin level. Let's quickly move on and look at pachytin once again. So the 9 sister chromatid of paternal and maternal chromosome, they overlap each other and appear to join at several regions along their length. So these points are normally known as the chiasmata. It is at this same chiasmata that's where now crossover will start taking place. So zygotin is responsible for formation of the bivalent, and that bivalent will lead to the process called synapsis. But there is another stage of prophase 1 where there is where it is called pachytin, and during pachytin, that's where you need to see the non-sister chromatid overlapping each other, joining each other at a point called the chiasmata. It is at this point, at the chiasmata, that's where you need to expect crossover to take place. That's now where the beginning of genetic variation starts from. So it is during this stage of cell division, that's where you and your sibling look different at this particular point, depending on what you inherited at that stage. So if you inherited if the sperm which fertilized the egg, it is the one which was carrying a tallness gene, that's why you are tall. If maybe the egg and the sperm, both of them were TT, and then you get married to another person with shortness, both of you are short, you might never have a child who is tall. You have short ones because you both have two small TTs. Okay, because of what you inherited. Is something that you cannot change. But let's say maybe the egg which was fertilized during that period was having a tallness, and the sperm which fertilized was having a small t, which is shortness, but the small t is dominant. Okay, that's where the case may be your mom is tall but your dad is short, and then you find yourself you are short. Okay, maybe it's because the shortness for the sperm which was released at that particular point was dominant or is dominant as compared to the tallness which is in the female. That's why you are short. But when you give birth, because of that tallness, that tall gene you got from your mom, and then maybe your husband is tall, and then you collide, you are likely to have a tall child. So there are certain things in life, in a human being, which a person can choose, and things that a person cannot choose. So my advice is, if you see someone, maybe they are dark, they are light, they look whatever, please don't laugh at them. They did not make such a choice. No one chooses to be what they were born to. Yeah, that's life. You can't choose to be born, and you can't choose to die. It's something that happens naturally. Okay, so exchange of gametes will take place at chiasmata. So still on packaging, and each chiasma is a site for exchange of genetic material between the chromatide. Remember, these are non-sister chromatide, and this process is called genetic recombination. It is known as genetic recombination. Furthermore, if we go there, we can now look at diprotein. So diprotein, the chromosome, they start to separate, and then separation is called disjunction. I'm much interested in this stage again. During this stage, that's where some complication come in. Okay, we have Down syndrome. Okay, we have Down syndrome. We have many other diseases that are as a result of genetic disjunction at this particular point. Okay, there are certain conditions, like I've already mentioned, Down syndrome, we have Patau syndrome, we have Edwards syndrome. All these syndromes I've talked about, they are due to the genetic disjunction. Sometimes, actually, most of the abortions, they take place due to genetic complications. Okay, due to if the chromosome which has been inherited, okay, if the chromosome which have been inherited, it is not in line with the growth, nature itself, it is able to have what we call a spontaneous abortion. That's why it's very important. Currently we have advanced technology. Before you get serious with someone before you settle. Go for the genetic checkup. Let them check what is your genotype, what is his genotype. Some people, because they don't check that, they end up having maybe a child with Down syndrome. They may end up having a child who cannot survive, whereby the woman will get pregnant, 2 weeks later the pregnant is lost. Maybe 3 weeks later the pregnant is lost during the trimester. So this is because of this stage, but I'm not saying this is the only reason. There are many reasons that lead to that. So this is something that you need to make sure that you are aware of as you make your decision as an adult. So many of the genetic diseases, they are as a result of this particular point, whereby there is no correct separation. Remember, this disjunction, it can lead to the formation of a monosomy or a polysomy. Yeah, a monosomy or a polysomy. Okay, so trisomy should be trisomy, either polysomy or trisomy. So this disjunction can lead to that, and mostly genetic disjunction, it normally takes place between the chromosome 13 to 21. In between these guys, that's where you need to expect some disjunctions to take place. Yeah, it's natural. Then the last one is dikinetics. This is where the separation is complete, and then with the paternal and the maternal chromosome having exchanged the portion of chromatide, and normally at this stage the chromosome condenses again, and then the chiasmata disappears. That's for prophase 1, and this is the chiasmata. So these are two homologous chromosomes. So this is an homologous chromosome and this is an homologous chromosome. This one and this one, they'll come together and they are going to form that chiasmata. So at this point, this is the zygote level. This is the zygote level, and then once they form a chiasmata, this is known as the packeting. This is known as the packeting level. So there they'll even do some exchange of genetics, and if you see these are non-sister chromatide. Okay, non-sister chromatide are the ones which are exchanging the genetic information. Then after the exchange, you can expect some genetic variation, and at this particular point they have separated, which is dikinesis. Okay, so with that, I can quickly look at metaphase 1. I don't know if you guys have a question on prophase 1. Do you guys have a question?
No.
All right. Okay. Since there is no question, we can move on and look at metaphase 1. So the rest of the stages are simple. They are very simple and you can easily understand them. So let me look at metaphase 1. So metaphase 1 has a short duration, and normally the chromosome move to the equator of the cell and come to lie in two parallel metaphases. and then the two parallel metaphases are formed by one set each of the homologous chromosome. Then there, each homologous chromosome has the kinetochore, one for each of its two chromatids. After metaphase, we need to expect the anaphase, and this is similar to anaphase in mitosis, except that each chromosome consists of two chromatids that remain held together. So here the chromatids remain together in anaphase. So in some exam questions or test questions, they will normally ask you to know the difference between anaphase in meiosis and anaphase in mitosis. So in meiosis, the chromatids are held together, but in mitosis they will surely separate. Then the chromosome, they will start to migrate to the pore, and then actual reduction in chromosome number occurs. However, each chromosome found at the pores consists of two chromatids. So a haploid number will be formed at anaphase. That's why you need to expect a haploid number to be formed at anaphase. But one thing you need to understand is that the two chromosomes, they normally have two chromatids. That's a very important point. So telophase, here now the chromosome at each pore, they normally uncoil, okay, and they elongate to form the chromatin. So nucleus reappear at each pore, and spider fibers disappears and centriole split. Then the nucleus membrane is formed at each pore, resulting in the formation of two daughter nuclei. So these two daughter nuclei which have been formed here are the ones that are now going to do what? Are the ones that are going to separate once again. Then we have the last stage of meiosis 1, which is cytokinesis. So during cytokinesis, there is the simultaneous formation of two daughter nuclei, and then a cleavage furrow appear in the middle of the cell. So this is a cell, like let's say this is a cell, and then there is a furrow which will appear in between them, and it is this furrow which will deepen and separate the two cells, the cell into two. Remember, each cell will have a nucleus of its own. Each cell will have a nucleus of its own, and it is these two cells which will further go and undergo meiosis 2. Okay, so for meiosis process, you need to understand that there is no interface between meiosis 1 and meiosis 2, but there is a brief intervening period which is known as interkinesis. Okay, so we have the stages of meiosis 2, which are just similar to those of mitosis. So I gave you an assignment in the previous class to research about what mitosis is. So these guys, they are just the same. You have prophase 2, metaphase 2, anaphase 2, and telophase 2. But meiosis 2 is similar to mitosis, the process of mitosis. So meaning that if you worked on that assignment, then you know what meiosis 2 is and what mitosis is. And then meiosis is normally begins soon after the completion of meiosis 1, following a brief interkinesis which has no DNA replication. So the meiosis 2 is meant for separating the two chromatids of each chromosome, and then the sister chromosome are portioned out amongst the two daughter cells formed in meiosis 1. So this is just the same thing which was in meiosis, mitosis, which I know you guys had to make some research. So the two daughter cells will result in the formation of the four daughter cells, and each daughter cell has unique genetic information. So you, your brother, your sister, or anyone, you have different genetic information, but you might have some small similarities, but still there is a difference in terms of almost everything. So when you talk of clinical application, we can talk about Down syndrome. I'll allow you guys to go and make some research how Down syndrome occurs. So there is what we call a chromosome translocation, which is normally defined as a genome abnormality in which a chromosome breaks and either the whole or a portion of it reattach to a different chromosome. So there are moments where a chromosome dislocates, okay, where a chromosome breaks. When that chromosome breaks, it's going to go and attach itself on another chromosome, and that is known as the chromosome translocation. This normally occurs between chromosome 13 to chromosome 21. I think this marks the end of our class for today's class. I can see some questions there. So this marks the end of our today's class. Those with questions, you can ask now, and those who are going to have questions later on, you are free to WhatsApp me.